Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs. Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond. They become very good at what they do, but only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.
This is Richard Jacobs with the Finding Genius Podcast. I have a very esteemed guest, Dr. Azra Raza. She's the Chen Soon-Shiong Professor of Medicine and Director of the MDS Center at Columbia University. She's a practicing oncologist that sees dozens of cancer patients weekly. She's been doing this for years and years and years. She directs a basic cancer research lab with hundreds of original publications in high-profile journals. She has a book out called The First Cell on the Human Costs of Pursuing Cancer to the Last that came out October 2019. It's a good book. I started in on it. It's an emotional book, but a very good one. And her life is devoted to early detection and prevention of cancer. So, Dr. Raza, thank you for coming.
It's an honor to be on with you, Richard.
Well, I know you've had to tell the story in different facets many times, but how long have you been working with cancer patients, and what got you into it, and what started you out?
That's a good place to start. I grew up in Pakistan, Richard, and was very interested in just nature in general to begin with. Very curious, asking lots of questions all the time. And when I was little, I would be following ants around, for example. And when a choice of career came along, there was no question in my mind that I wanted to study science, and especially natural sciences. But the problem was that in Pakistan, we did not have advanced science degrees except through medicine. So that's why I did medicine. But once I was introduced to my first patient, that was about it. I knew that from that moment on, everything I do would be in service of patients.
Okay. And you've been working with cancer patients for how long, 30 years?
30 plus years now. So I came to this country in 1977. From that point on, 1980s, that's all I've been doing.
Oh, wow. Well, this is part of the problem, but, have you seen cancer treatment change much over the past 30 plus years?
Dramatically change. For example, when I came in 77, I was treating acute myeloid leukemia, the disease I began by studying and treating with two drugs called 7 and 3. They're called 7 and 3 because 7 days of one and 3 days of another. And they were only 10% patients who survived 5 years as a result of this treatment with the two drugs. That was in 77. Today, in 2020, on the one hand, we are still using the same two drugs, 7 and 3. But on the other hand, the survival is 50% in 5 years. Why? Because we learned to take better care of patients in terms of better antibiotics, better supportive care, anticipating problems, giving bone marrow transplants. So in some ways, it's dramatically different. In other ways, it's embarrassingly, shockingly primitive.
Seems to me the understanding of cancer, the understanding we think we have, is fundamentally flawed. From what I've learned in speaking to a lot of researchers, that cancer is very heterogeneous. There's not just one cell. There's many, many different kinds. And, it's a living thing is what it appears to me. It has its own defense mechanisms. It has a tremendous set of abilities to adapt and change, and hitting it with just one or two drugs, especially in light of the fact that it doesn't really seem to help, and it just emboldens it to come back more aggressively and worse. I mean, it doesn't seem to sink in. A different approach is needed, it seems like.
Richard, what you're saying is not anything that comes as a surprise to anybody. We have all been saying the same thing forever. Since I landed in this country, this has been the conversation, that cancer is heterogeneous. It's not one disease in the same patient at two time points or at two different sites. It's not the same disease. The question is, why can't we rise above all this then? And something that I'm very allergic to is that younger people come in waves, acting as if the last 60 years haven't existed. Do you know that my book, The First Cell, was being discussed by a class of Yale students? And so last week, their teacher asked me to join.
Okay.
And I was asked a question, something like you asked just now, that cancer is so complicated and, what's a better way of dealing with it than just slash, poison, burn, trying to hit it with everything we have and get rid of it in one way or another, cut it out or poison it or burn it. What else do you think? And so I started to tell them about how complex it's not just the cancer cell itself, its genetics, its microenvironment, the blood vessels that are supplying nutrition to it, the cytokine storm all around it, the immune cells trying to control it. It's so complicated. One of the students said to me, well, Dr. Raza, that may have been for you, but right now I'm working with a scientist and we understand that in order to successfully treat this disease, we have to take evolution into consideration. And so we are designing therapies that will attack simultaneous waves of cells as they evolve. To this, my response was, the hubris involved in thinking that no one else has thought about this first. In 1976, Peter Noel wrote a classic paper, which has now become a classic paper, describing the clonal evolution in cancer cells and predicting exactly the conundrum we find ourselves in today. And we have painted out the corner with over and over that each time a cancer cell divides, it picks up new mutations. So it's giving birth to 2 new cancer cells potentially, and that there will be no way to deal with as this advanced kind of disease. The simpler, the better. The earlier you get it, the better it is. There's no point in keep hacking at the leaves. You have to go for the root, etc., etc. This is back in 1976. In 1977, one of the first papers I reviewed in the lab was this one. And it laid out the entire story, which has only been confirmed word to word by all the fancy genomics and transcriptomics information. And I had to tell this young man that you can't act as if the past doesn't exist. I was, in fact, one of the first people who designed with my late husband the protocol, which was called timed sequential therapy. This is exactly what we were doing, Richard. Can you believe it? Even the names, timed sequential therapy. I was doing it in 1977, which this young man is lecturing me about now.
So even giving cancer I don't want to say the respect it deserves, but it's amazing its power. Do you think that there is any way, once it advances beyond a certain stage, that we can fight it? Or is that just because the battle's gone on for so long, are we out of ideas? Is it really only catching it early that you think is going to be the thing that can stop it? Like, what's your overall thoughts?
I think those are very good questions. My overall thoughts is just look at the field as it exists today. We are curing 68% of the cancer, and that's great. But what are we curing them with? Slash, poison, burn, surgery, chemotherapy, radiotherapy, the same treatments we were using, save for a few rare exceptions. So I don't want people to think that I don't know about antibodies or immune checkpoint inhibitors or CAR T cells. Those are very rare exceptions. By and large, the common cancers are treated with the same traditional therapy. It is shocking that in this day and age of such advanced technology, we are using such Paleolithic cavemen treatments. These treatments were given basically as stopgap measures. We are going to understand the biology of cancer and then try to undo the damage that has been done to the cell to convert it from normal cell. And hundreds of thousands of researchers in the last 50, 60 years have worked very hard to do just that. And we have not succeeded. Why? Because it's a moving target. Sure, we are curing 68%, but the reason for the fall in mortality by 1% a year in the last 3 decades is only because of early detection and reduction in smoking. So the earlier so it's not just the earlier the cancer detected, the easier it will be to treat. This is what history is showing us, that the only way we are making any headway is even the same very effective therapies that cure 68% fail entirely when the disease is advanced, the same disease. So if early detection seems to be the best way right now, while we work out the complexities of metastatic advanced disease, then why not go for proper early detection using all our resources?
You know why I think part of the reason is that when I ask people, how does cancer start? The story is just, we don't know, or, one cell randomly mutates and then it starts cancer. And I just don't think that is how cancer starts. And I think if you view cells as machines and not living organisms that adapt and have some level of cognition, and, if you view the beginning of cancer as just some random event, I think that's going to make it a lot harder to actually figure out how it starts.
No, I mean, I can't argue with you whether it's a random event or an adaptive event. Adapting to old age, for example. There are causes of age are poisoning the environment, making cells misbehave. Every morning I get up and look at the mirror and it's shocking to see myself. But the thought that comes to mind is, if this is what age is doing to me on the outside, imagine what it's doing inside. So you're right. It's a very simplistic way of thinking about a cause of cancer, or one cell going rogue, or one mutation causing, wreaking havoc on the system. It's all these things that are co-evolving simultaneously. And this is why cancer is so much more common. The immune system is becoming decrepit. The cells are collecting more and more mutations. The systems around in the microenvironment are not as efficient in removing damaged cells and garbage disposal as they were before. So the whole microenvironment is becoming poisonous and only supports the growth of a cell which is slightly got a growth advantage due to some mutation or another. I think you're absolutely right. It's misplaced to think of it as one cause.
Well, if you're saying early detection is going to be critical into making progress in cancer, then we need to really focus a lot on how cancer starts, I would think. Otherwise, how are we going to know how to detect it early if we don't know the hallmarks of it when it first starts?
I don't think you need to know how it starts. What you need to know is what are its footprints in the earliest stages. Evolved from a benign pre-leukemic form to a more malignant stage. And do you know that in these 60,000 samples, Richard, not a single cell comes from another oncologist? I still do the bone marrows.
You do them all yourself? Wow.
I still do the bone marrows myself. They are all my patients. Every vial, every cc of blood or marrow has a story behind it. And that's why I am all the more responsible that patients underwent a lot of pain, but they trusted me. Dr. Raza, even if it doesn't help me, if you think it's going to help someone else, go ahead, take as much. Imagine the grace and nobility of somebody who knows they are dying, but who are ready to help others. But there are no grants available to do it. Do you know who supports me? My pay over $30 million by itself, but they are doing it because it's to their benefit also. And to my benefit. So this is another way of doing it. I'm trying my best to get the same thing done for proteomics and for transcriptomics, because I don't see anyone coming in and giving me the resources to do it. I can't tell you how many times I've written to the NIH, by the way, to the director, myself, so many times over the years. They have no interest at all.
Why?
This is what shocks. Why? Because, the thing is that most of the decisions we make are not made because we want to make them. That's what we want to do. In fact, most of us oncologists do things we detest doing, but we have to do them because they have been defined by key opinion leaders who say that if somebody presents with pancreatic cancer, then the first line of therapy is this, the second line of therapy is this, third line is this. If I deviate from that, then I'm opening myself to legal challenge. Why isn't the NIH director interested in supporting this huge tissue repository and helping me study it? They don't have $100 million. That's very simple. Our budget is very small. How do they support it?
Well, if you made a Pareto of the first experiments, well, I know you're doing some already, but
study. But the way I'll end by saying the way the system has evolved in this country is that, let's say I, as an individual scientist, can apply for grants to the National Cancer Institute. Let's say I get the grant for 5 years. It's $250,000 a year for 5 years. With that, I can find one target and say, oh, I found a target. I think drugs can attack this target and will cure. Announcement from ASCO saying, I won't name names, but saying that a drug has been approved for ovarian and cancer of some sort, in which, look at the insanity of this. The drug is approved because it improves by 5 months against the placebo, improves progression-free survival, not survival. Can you imagine that we have come to the level where we've given up on improving survival in patients? As long as you don't see the tumor, that's considered a response. Because the tumor is microscopic, it will kill in the exact same time as it, because there's progression-free survival. Why are our agency in that? Yes, earlier and earlier is the only way we have found anything historically.
You know why I ask? Because I hear a lot, early detection, but no one defines why that's better or what that means. That's the problem. I've heard that a lot. You have to catch it early, but again, I just haven't heard an explanation of why that makes it better.
This is why, because it's less complicated. Each time a cell will divide, it will become more complicated and more adapted, as you said, brilliantly to its environment, because that's the only one that will survive, one that is showing its fitness to the landscape. So more and more complicated cancers emerge, which can be resistant to all kinds of treatments. But if caught early enough, they are more vulnerable. And this is what has been shown over and over. In 1908, Dr. Childy said, it's not cancer that kills. It's the delay in treatment that kills.
What's the earliest cancer that's ever been observed, or earliest set of cancers? Like, what do they look like? How do they look differently than later ones?
The kind of thing I'm going for, no one has any idea. Because what I'm saying is that, let's say, and this is all happening, this is all being studied. Let's say I will go to sleep in bed sheets that will scan me overnight for the appearance of a hot spot in my body anywhere. What does that mean? Well, as soon as cancer begins, it has to feed itself because the cells are dividing more rapidly. So it starts attracting more and more blood vessels to it. So the area becomes hot, starts forming new blood vessels. And that can be picked up easily. And so let's say that a bedsheet detects that there is a hot spot in the head of my pancreas one night. That doesn't mean I wake up in the morning and should have an open abdominal surgery and evisceration of all my organs. All it means is that there is an area of a region of interest in the head of pancreas. Now it should be followed more carefully with all the imaging, scanning. We have artificial intelligence. We should be using that to hone in and direct our attention to that particular area, but then try to supplement it with 20 other tests. Try to look for what does a common pancreatic cancer shed as proteins in the urine, in the serum, in the plasma, in saliva, in the breath. What are these proteins that we can catch? So we should not depend on a PSA or a colonoscopy. We should be having 20 tests to supplement that one finding. And once over a period of a month or 2 months, we realize that hot spot has stayed, is growing, is getting hotter. And now we are able to detect these proteins even in the urine and serum and saliva, then we know that whether it's really potentially lethal for the individual or not. Should we go after it or not? And if we have to go after it, then it's a tiny little blip right now in the pancreas. We can probably just use a well-directed super laser beam to just zap it out of existence, instead of trying to give radiation therapy for 2 months. So treating diseases to preventing diseases, just like cardiovascular people did. Look, abnormality.
What's I'm not trying to be snide here, but what about the microbiome? It's not really new, but it seems to be new and not very well considered. Do you think that's going to be a very important part of understanding cancer? I mean, the reason why I ask is this: I interviewed a lady named Florencia McAllister, and she was studying pancreatic tumors. And she said that the tumors themselves had a localized environment, that it was a different microbiome than the rest of the pancreas. So perhaps that will be a very important thing, is to look at the microbiome of the affected organs or tissues as well.
Yes, I know this work very well, and absolutely, those are the things we need to study. Instead of studying mice, we should be studying microbiomes in humans and trying to determine how that signature changes as cancer develops or evolves. I think all of this works in concert. It is the right thing. Yes, and you were not being snide. You're being very brilliant by asking this question, and very knowledgeable.
In regards to your tissue repository, are you still trying to grow it? Because